Hello friends, I am Vineet Sharma. I am a professor at IISER Bhopal in the Department of Biological Sciences. In the January to April 2024 semester, I am going to teach the Computational Genomics course in the NPTEL course program series. So today I will give you a brief introduction of what I am going to teach. So it is right to say that we have arrived in the era of big data of biology, which has created a lot of new computational challenges. You can imagine that in the year 2000 it was like the NCBI reference database was just 1 GB, but today it will be like more than 160 170 GB. So and the commonly used sequencer are producing so much data that in 2 days, which in a simple PC you are going to take almost 500 years to analyze. So that is why you really need good computational skills to analyze this large data. And not only this, there are so many other projects all over the world, such as the Human Microbiome Project, the Bird Genome Project, the Genome 10K, Earth Microbiome Project, where they want to sequence everything that is there on this planet. They are generating a lot of data that needs analysis and also people to analyze these data, which are even more important. And it all started, as you know, that with the discovery of the DNA structure by Watson and Crick, followed up by the invention of the sequencing methodology by Sanger, which we commonly use today. That led to the evolution of the field genomics, which include the study of the structure, function, organization, sequencing, evolution, mapping, editing of genomes. So it's a complex and multifaceted area. And the prominent genome that I must mention is the human genome. That was 23 pairs of chromosome, 25,000 genes, 3 billion base pairs, and basically it was first done by the Sanger Reeds, which was in bits and pieces, and it was published in year 2000 and 2001, okay, almost 20 years back, right? So, and today, like if you see that, it took almost 20 years to complete this genome. So imagine that all the scientists all over the world, so many computational scientists are working all the time, investing their money, but still it took so long to complete the human genome. That means that this area is really difficult, and it involves time and skills to complete a genome. So in this course I am going to teach that, like, with the availability of the large amount of sequence data from the genome, transcriptomes, epigenomes, metabolomes, proteomes. So basically we need skills in students, researchers, faculties to handle this data, to ask questions and analyze this data. And basically that is why in this course my emphasis is to build concepts and skills in the area of computational genomics, and I will provide you insights into what goes into a complex genome analysis, okay, and we will look at the various algorithms and methods to do this in the next 12 weeks. So in the week 1, we will first see that what is genomics, what do you mean by computational genomics, transcriptomics, proteomics, epigenomics, metagenomics, what is the big data of biology, then how this information is stored in the complex prokaryotic and eukaryotic systems, and what methods are used to generate this information. Then we look at that how this data is actually organized in the public databases, which you can retrieve and use for your work. Then in the second week we will look at the different sequencing methodologies that are used to produce this data, including the traditional and the new age methodologies, and also like how this data is stored in the various sequence formats. In the week 3 we will learn the concepts in Linux R, which is very important to handle the data, because you have to use a Linux system to address and analyze this data. In the 4th week we will look at the commonly available packages such as Emboss and others to do this genomic analysis. We will also look at how in the past studies the genome and transcript analysis has happened using the case studies. That will help you to build a new project, right? So in the 3rd class in this week we will look at how to design a project, including sample collection, DNA extraction, and other things that goes into designing a project. In the week 5 we will look at how to estimate that how much data you should produce and how to analyze it, okay. We will also estimate the complexity of the genome and various parameters that are important for it, and we will also look at the concepts of genome assembly, including scaffolding, complete genome, draft genome, etc. In week 6 we will look at various assembly methods such as ABASE, SOAP de novo, FLY, etc., which are used commonly for the genome assembly. right, including the link reads and the long reads such as nanopore. In week 7, we will look at the methods that are used to assemble a genome, polish a genome, and to generate a assembly that you can report for publications, including the various parameters such as N50, boost score, etc., which are important for it. And also we will look at Hi-C, which is important to complete the chromosomal construction of a genome. In week 8, we will mainly deal with the annotation of genomes, including the repeat annotation and the genic prediction annotation, and also the non-coding RNA predictions. In week 9, we will spend time in how to annotate these repeats, genes, and the other non-coding genes that we get, and also do their comparative and evolutionary analysis, and what are the methods for the various phylogenetic and taxonomic analysis for the data that you have already produced from your project. In week 10, I will introduce you to the epigenetics, ChIP-seq, transcriptomics, etc., which is very important these days, including the single cell genomics, spatial transcriptomics, and also the various other multi-omics approaches that are important for human health and disease, which are such as cancer, diabetes, etc. In week 11, we will focus on the prokaryotic genome sequencing as an assembly analysis and annotation of the various things that come out of the genomic analysis, and also the phylogenetic and metabolic reconstructions. And in week 12, we will spend time on the microbiome and metagenomic analysis because these are very, very important, these areas these days, and their annotation, and also the taxonomic and phylogenetic analysis of the various microbiomes and metagenomes, including the statistical analysis. So it's a very, very comprehensive and complete course which is going to develop concepts in genomics, transcriptomics, epigenomics, and metagenomics, including Linux RR in those who are taking up this course. And I think it will be a very important course to learn the skills for the handling the large amount of biological data and analyze it, and including the insights, algorithms, and the commonly used techniques and platforms that you can use for it. And this will also help you to design your own projects. Okay, so that is why I strongly encourage you to register for this course, which is named as Computational Genomics, available at the NPTEL portal. And I look forward to seeing all of you in this course. Thank you.